Hello and welcome to this evening's UCSF Virtual Alumni Program, Pivoting to Fight COVID-19. Thank you all so much for tuning in and for being a part of such a special virtual experience. My name is Serena Tamura, and I'll be your moderator this evening. I'm currently a graduate student here at UCSF in Nadav Ahituf's lab in the Department of Bioengineering and Therapeutic Sciences, and I'm also a PhD candidate in the Pharmaceutical Sciences and Pharmacogenomics program. I'm also a UCSF Discovery Fellow, an Autism Science Foundation Fellow, and I continue to volunteer at the Chan Zuckerberg Biohub UCSF COVID Testing Lab. Now, before we begin with our program, I'd like to go over a few housekeeping items to help with your experience. To submit a question during the presentation, please use the Q&A box. And if you could, do your best to keep your questions as brief as possible. And given the size of the audience and just the number of questions that we're anticipating, our speakers may not be able to answer all your brilliant questions, but don't let that deter you. If you do have a question, go ahead and post it in that Q&A box. I am so excited to introduce you to three UCSF alumni whose work has shifted quickly and importantly to focus on fighting COVID-19 earlier this year. We're going to hear from each of our speakers and they're summarizing their experiences, and then we'll open it up to your questions and have a conversation on the lessons they've learned and what they see as solutions to finding the best testing, the best therapeutics, and perhaps ultimately a vaccine. Our first speaker is Jason Fernandez, who received his PhD from UCSF in 2013 and is also a postdoc alum. Jason completed his doctorate studying the evolution of HIV in Alan Frankel's lab at UCSF. He studied the evolution of viral overlapping reading frames in HIV, as well as virus-to-host protein-protein interactions. Jason is currently a project scientist at UC Santa Cruz's Genomics Institute. UC Santa Cruz Genomics Institute, where he studies how viruses and other genomic parasites have influenced primate evolution. Welcome, Jason.
Thank you, Serena. As was mentioned, I'm a graduate alum at UCSF, and I did my PhD in Alan Frankel's lab, where I studied the evolution of HIV. And I also collaborated with Nevin Krogan's lab, mapping HIV human host protein interactions. And some of you are probably familiar with the amazing work that Nevin's lab has now done to take that technology to use in the fight against SARS-CoV-2, the virus that causes COVID-19. So after my time at UCSF, I went to do my postdoc with Dr. David Hausler at UC Santa Cruz. And at UC Santa Cruz, I got to work with the UC Santa Cruz Genome Browser team. So that's the team that assembled and published the first draft of the human genome. And so this is in the genomics field, this is a really highly accessed resource and sort of the most common way people interact with the human genome. And while I was a postdoc in David's lab, I was still interested in viruses. And I was sort of studying how viruses and virus-like sequences have really shaped the human genome. And I was doing good and important work on that. But then I think, in January, as you all know, the world kind of changed. And at least on the genomics side, that's when we first got the sequence for this novel coronavirus, SARS-CoV-2. And so when that genomic sequence was first released, our team sort of went ahead and did what we typically do, which is release that full genome sequence as what we call a genome browser. So we built a SARS-CoV-2 genome browser. So to give you an idea, that genome is only 20 or 30,000 nucleotides long, whereas the human genome is 3 billion. So it's a much smaller, more compact thing. And typically when we build these browsers, then in addition to just having the nucleotide sequence of the genome, we then sort of overlay that with annotations, things like where does a gene start, and then other maybe interesting experimental information like RNA secondary structure, immunogenic portions of the genome that might be potentially immunogenic, those sorts of things. And we've done this in the past in previous sort of viral outbreaks, for instance, in Zika and Ebola, and released browsers. But while there's maybe been an initial sort of burst of interest, things have really sort of quieted down as those outbreaks have been controlled. And obviously that's not the case here. With SARS-CoV-2, it's unfortunate that we haven't been able to sort of get a handle on this virus as quickly as we have with some other outbreaks. But the sort of flip side of that is that the pace of the research has been really astonishing. And every day new groups are releasing a lot of different molecular information on the virus. And so what we do at UC Santa Cruz is we transform that information, that sort of molecular information that tells us some sort of feature of the virus that corresponds to a nucleotide, and annotate the genome in a way that people who do big data analyses in file formats that they're used to working with. And so that way they can do these kinds of analyses using sort of proven approaches. And so if you go on the SARS-CoV-2 Genome Browser today, you'll see annotations for immune epitopes. You'll see alignments to other coronaviruses, to other human coronaviruses, other bat coronaviruses. And so you can sort of get a lot of information and do really big data analyses. And so just a very short vignette about the kinds of analyses we were able to do on our own end is, as we were building this browser and sort of organizing this information, we noticed, and it was great that so many labs were sort of sharing the genomes and sequences of viruses that were sort of being seen across the world. We did notice that several mutations that were appearing in the virus did not appear to be actually follow the predicted pattern of evolution, that they didn't seem to be real mutations. They seemed to follow a pattern that seemed to be more like artifacts, maybe sequencing or assembly errors. And using a bunch of different methodologies, we were able to sort of show that this was in fact the case, that there were certain sites in the viral genome that weren't really amenable to being sort of assembled correctly depending on the approach. And at the same time that we were doing this, scientists in the UK and Australia were noticing the same thing. And so one, that kind of reassured us that we were on the right track and seeing the correct thing, but we were all able to come together and then release a sort of file with sites in the genome that sort of are warnings of areas where if you think some interesting biology is happening at this point, perhaps you better double check that that it is truly interesting biology and not an artifact of the methodology. And so it was really reassuring to sort of see other groups arrive to that. And then it was really great for us to all come together and collaborate on it rather than rushing to get a paper out. So I guess if I could just leave you with sort of take-home messages from my experience, is that I've been really astonished by the pace of the research. And it's been tough on our end to sort of organize and sift through all the data as it comes out. But then I've also been excited by the sort of collaborative effort where people, it's not entirely, there have been a couple of places where I wish people would be a little more open with their data. But in general, people have really been trying to figure out how we can stop this virus as opposed to, advancing their own scientific careers or their own sort of glory of discovery. So I think it's been overall a very positive experience. And so that's been sort of my story and sort of pivoting to COVID research. And so I think now Serena will introduce you to Emily.
Great. Thank you so much, Jason, for sharing your incredible work with us. And I use the UCSC Genome Browser every day, so it's awesome to get an insider's scoop. Please submit your questions for Jason in that Q&A box, and we'll work to answer them shortly. Next, I'd like to introduce Emily Crawford, who received her PhD in 2012 and is also a postdoc alum. Emily carried out her PhD thesis in Jim Wells's lab at UCSF, where she studied the evolution of caspase activity in the programmed cell death of a wide range of metazoans. As a postdoctoral fellow in Joe DeRisi's lab at UCSF, she devised CRISPR-Cas9-based tools for depleting abundant sequences and enriching rare sequences in next-generation sequencing libraries. Emily currently serves as a project leader at the Chan Zuckerberg Biohub. Welcome, Emily.
Hi, thank you, Serena. Thanks, Jason. That was really, really interesting. I also really enjoyed the UCSC Genome Browser. It's great. And Serena, thanks for moderating this, and also thank you for all of your hard work working with us at the CLIA Hub. My name is Emily. I am a biochemist by training, and as Serena mentioned, I'm a group leader at the Chan Zuckerberg Biohub, which is a nonprofit research organization that's located on the Mission Bay campus and has affiliations with UCSF, Stanford, and Berkeley. So we work very closely with a lot of UCSF labs, and in particular, my research since finishing my postdoc in Joe's lab has been on developing new ways to use CRISPR-Cas9 and new ways to use metagenomic sequencing for infectious disease diagnostics. I worked closely for the past few years with the clinical microbiology lab at China Basin at UCSF. We did a number of projects with them, looking at outbreak investigations, hospital outbreaks and things like that. And as a result of that, I think we were a natural partner for the clinical microbiology lab back in March, when the decision was made to try to expand the COVID-19 testing capacity of UCSF by taking advantage of the amazing resource that we have in the form of graduate students, postdocs, assay development expertise, equipment, everything that we use in the research labs that we don't normally put towards actual clinical testing, but that we could and we do now. So it was back in early March, I think March 12th, was when the governor issued an executive order altering a little bit the requirements for performing CLIA-approved diagnostic tests in California for COVID-19 specifically. So whereas previously there still are, and for everything other than COVID, restrictions about CLIA labs needing to be associated with a particular street address, and restrictions about the types of certification needed for people who are performing testing. And these were relaxed a little bit, and that allowed UCSF to call up the Biohub and ask us if we would take on the project of spinning up a COVID-19 testing facility. And we did that. It seemed like the most craziest idea I had ever heard of at the time. It still seems a little crazy because I have absolutely no clinical background. Everything I've done in my career is purely research, and that's the case for the vast majority of people at the Biohub. But we recognized that this was something that was needed, and we had been sort of mulling over, should we be doing some research studies, and if so, how are we going to get PCR testing done for in a research type of setting, and would it be ethical to do it while not reporting the results back to people because it wasn't a CLIA approved test, et cetera. So this decision by the clinical microbiology lab to ask us to spin up our own lab sort of made all of those concerns go away, and we realized the best thing we could be doing at the moment was actually providing increased testing capacity in the form of a PCR, RT-PCR, LDT that we would build up. So we gathered a team of people at the Biohub who knew how to do assay development, knew how to run PCRs, how to do RNA extractions, how to use liquid handling robots. We actually solicited from all over the Bay Area, all sorts of different companies and labs and universities to get additional liquid handling instruments and things like tips for our Agilent Bravos, RNA extraction kits, et cetera. We were just inundated with people wanting to donate equipment and supplies. And that was really nice. And so we set up a facility where we have 8 Agilent Bravos. We have a couple of Hamilton Starlets. And these are liquid handling robots that just help us to do the RNA extractions and qPCR. And to staff the facility, we were happy to realize that there were hundreds of grad students and postdocs who were, sitting at home in lockdown because they couldn't access their labs, or they had very limited access to labs. And Serena was one of those. Thank you again for that, Serena. We had a couple hundred people volunteer to help us out. And so we started taking them in and doing training. And over the course of a few weeks, we developed a workflow and figured out how to do these tests. All as sort of acting as a part of the clinical microbiology lab at China Basin. We're overseen by Steve Miller and Charles Chu there, and nothing that we've done could have been done without them. Let's see, we've now processed 115,000 tests. That is about 1.5% of all the tests that have been done in the state of California. Thanks to our donors for the Biohub, Chan and Zuckerberg, we're providing free testing to the public health departments around the state of California. So every day we get samples from about 20 to 30 different counties across California, public health departments, most of them in the northern part, but we get some from Long Beach and SLO, and we're actually processing some Santa Cruz samples right now that couldn't be processed down there because of the fires. We're able to provide that free testing and have various impacts on what's able to be done in the public health sphere. We've also been able to provide testing for a number of different clinical studies that have gone on at UCSF, such as the Mission District study that people may have heard of. It was in the news back in April, showing an incredibly high proportion of positive people being Latinx people, and high proportion of people mentioning that they are unable to work from home, sort of showing us what kind of populations are most strongly hit in this crisis. So yeah, we're still operating. We now have staff. We've hired about 12 people. So we're not exclusively relying on the volunteers anymore, but we are still relying on them to train the staff. So it's really great to still have so much help from them there. And I think this is really this has been an experience, unlike anything that I've ever been involved in before. And I really think it's something that's very emblematic of UCSF. I don't know if this could have been done anywhere else. I think this is really, it's an unusually strong collaboration between basic science and clinical science or clinical diagnostics. That couldn't easily be done without sort of the kind of environment that we have here of being collaborative and helping each other out, and being creative about seeing the connections between things that might not be obvious. So I'm really happy to have been able to do this at UCSF. And I will end there and turn it back over to Serena.
Wow, thank you so much, Emily, for sharing your experience of starting a COVID testing lab from the ground up to 115,000 tests. That's amazing. And on a personal note, as a female trainee, I am so proud to have had this experience of volunteering with you at the CLIA Hub. Thank you so much for your fearless leadership. So please submit your questions for Emily in that Q&A box, and we will get to them shortly. Next, I'd like to introduce Alex Greninger, who received his MD in 2015, his PhD in 2013, and is also a postdoc alum. Alex is an assistant professor of laboratory medicine and the associate director of the clinical virology laboratory at the University of Washington. He earned his medical and doctoral degrees from UCSF, followed by a residency in clinical pathology at UW. His work generally involves genomically informed approaches to understanding a variety of infectious diseases. Welcome, Alex.
Hey, thank you very much, Serena. Yeah, so it's been great to hear the stories from Emily and Jason. I've actually left San Francisco in 2015, come up here to Seattle, and kind of got hooked up here in the clinical virology lab in residency. It's been a great experience. This is basically what we do, right? So I guess I wouldn't even call it pivoting to COVID response. I mean, you have to pivot in the sense the virus came out and there's something to be done. There wasn't a test for it before, but we really just sort of done what we do. I've always had a love for RNA viruses, so was really paying attention to what was going on in China at the end of December. Ordered primers basically as soon as the genome came out. We basically do a lot of clinical trials, so even if the thing never made it to the United States, we thought we probably would end up doing some clinical trials. Put it in R21 for coronavirus as well in the first week of February, just expecting sort of more interest in coronaviruses coming down the pike. We've done a couple of pieces on coronaviruses, mostly in our stem cell transplant patients. There's really, these viruses are notoriously underworked on the 4 normal human coronaviruses. By comparison, they're probably almost 100,000 genomes. Just as an example, we're very genomic focused, right? All 3 of us, but they're probably around about 90,000 genomes for the SARS coronavirus 2. If you add up all of the genomes for the 4 coronaviruses that regularly infect humans every year, you're probably talking around 600 total sequences in those guys. And it's just incredible how little people care about them. But they do cause problems in stem cell transplants. So we worked on them a little bit before. So we were attuned to some a little bit of coronavirus work before. A lot of the effort that we spent early on was, trying to develop a test and then dealing with the FDA through month of February. There's been a lot of lingering debate over FDA regulation of clinical laboratory tests and laboratory developed tests. And so, I actually took a lead in sort of interacting with the FDA, which is a first. We don't really interact with them that much, and trying to see what the process was. And then was involved towards the end of February with lobbying Congress to reinstate CLIA, which they did on February 29th. And we got a positive sample here in Seattle. For some reason, the virus decided to make Seattle its sort of one of its starting places here in the United States. And we were able to, through sort of some of that early work, engage with the FDA over the entire month of February, as well as the change in regulation and the fact that the positive control sample showed up here, we were able to start testing on Texas Independence Day, March 2nd. So that's how I always remember it. But we just, for the first 2 weeks, we were about, maybe, I don't know, we were one of the first labs in the clinical labs in the US to perform testing. We were somewhere outside of the public health labs, maybe a quarter to a half of US capacity for testing during those first 2 weeks. And within the growth in testing during that time was basically an exponential curve, and we absolutely crumped the second week of testing. We just basically exhausted our pre-analytical space. We went from doing, about 20 tests, 20, 30 tests the first day to about 3,000 tests within about 2 weeks. And there we had to put some, basically triage level, sort of tiering of samples, focusing on inpatients as other places are, and sent a lot of samples actually during that time around the United States to try and solve that positive control problem so other labs could stand up testing very quickly. That's really unique. We basically, as a lab, we do something like 50,000 tests in a year. One of our highest analytes, CMV, would be something about 12,000 tests a year. So at that time, even just running 3,000 tests a day is ridiculous. Now we're doing about 7,500 to 8,000 tests a day for COVID. We basically run our annual volume for molecular testing every week currently. So it really is remarkable what's happened with this virus from a reimbursement, from a public health, and just from a testing angle. People think that testing is actually going to change a lot of the issues here, but we just keep testing. Also during that time, basically in April, we started pivoting to some serology tests. We put out one of the early Abbott serology tests, and we're sort of helped in sort of righting the ship with sort of the bad serologies that had come out. The first 100, serology tests that have been registered with the FDA. The FDA had sort of overlearned its lesson at that point, and it was doing less and less regulation on serology. And then finally, in the end of April, some of the better tests came out. And then May, we basically just kind of like held pat, honestly. Our curve had peaked on March 29th and had gone down in April. We didn't have a lot of cases. It had sort of moved on to Yakima, and people were focused on other parts of the United States. Our allocations started getting restricted, even as we brought on more sample to answer instruments. And that's just what it was. We really didn't have much to offer. And then finally in June, the state decided to just start testing every single nursing home resident and that really kick-started very quickly. We're receiving just hundreds of boxes a day from different nursing homes, some of them not even packed at all. And then finally, drive-throughs got started again in earnest, and now we do about 3,000 samples a day from drive-throughs here in Seattle. And the county came through today to basically look at making even more drive-throughs. It's been where it's sort of worked the right mix for the testing regulation. It's been really hard to get at-home testing off the ground from an FDA regulation standpoint, and we've been able, and this sort of observed drive-through has worked well for us. It's one of our mainstays of testing. Emily talked about some of the staffing, now we've probably hired more than 150 people here, as well as during that first punch in March for different, grad students and people who were sort of at home as the labs closed down. We had a lot of help. Actually, Chris Nelson from UCSF, one of my grad student alums, grad student in the same lab, and Dorisi's lab came up here and actually just decanted samples for 2 to 3 weeks, which was really great to see, and some of that UCSF love. Recently, it's just been trying to deal with burnout on the staff angle, and then all the testing regulation changes that come up, and just maintaining that high volume of testing while trying to still bring on additional platforms, especially with the July peak through the United States. As many of the reagents got reallocated to Florida and Texas and California, Southern California for the most part, and just trying to go back to LDT, our laboratory developed test, back to sort of DIY type testing, focusing on pooling. That's been very helpful for us to be able to get a lot of tests out. And now you're seeing more and more push towards decreasing sensitivity testing to allow more widespread testing. I think Abbott just got an antigen EUA out today. It'll be interesting to see how that changes the testing market and the testing approaches and algorithms. I mean, it's just constant revolution. You just can't get any. You just can't get any regularity to it. There's always something going on. It's really, really exhausting. It's been hard on a lot of the labs here. We feel like we're at sort of like mile 15 or 16 of a marathon right now. And, they want us to do burpees, or they want us to do something, something special. Or they want us to everyone wants, especially at 7,000, these number of tests a day, you have all these edge cases that come up. That's been its own issue. So right now we've been very focused on the phase 3 vaccine trials and getting testing set up and working with a lot of different companies on those, very clinical trials. A lot of drugs actually worked on before in the basic science lab, down at another campus. But for the most part, it's been all COVID all the time. And happy to talk about anything from a testing regulation standpoint to the actual what we're doing, what we see in the future, and what's kind of going on here. Also from a vaccine angle, just happy to talk and understand what people are thinking about and what they're concerned about. It's been really true that the versatility and the full health sciences education, from the basic science side to the medical side to the to getting all of your CLIA ducks in a row, and again, I feel like I've broken record. I've become much more versed in regulation than I ever thought I would be during this time. With that, thank you very much for hosting, and I'm happy to talk any questions.
Thank you so much, Alex, for sharing your experience, not necessarily pivoting, but perhaps pioneering and diving even deeper into fighting COVID-19. If anyone has any questions for Alex, go ahead and post that in the Q&A box. I just want to thank Alex, Emily, and Jason for sharing your incredible work, for what you've been doing to fight COVID-19. I am so proud to be a part of the UCSF community. So let's go ahead and get to those questions that our audience has shared. We have our speakers visible again. All right. Let's see here. I guess this question might go for Alex or Emily. Any thoughts on recommendations for a reliable serology test, specifically IgG?
Yeah, so you want, I think the best test right now out there is probably the Ortho MCL anti-spike antibody tests. The main ones that have come out of the ones that are most widely used, if you go to a Quester LabCorp, are going to be an anti-nucleocapsid from Abbott or Roche, which have high penetrance and are pretty good. They're really good. But the ability to get that sort of, it's not really an RBD assay, but an anti-spike assay that can be run on a high throughput, something you can actually get and get sort of cheap and have an FDA EUA, I think probably is the best one so far. but it depends on different use cases. That's sort of, and then also the Beckman, was it Beckman? Beckman, I think, has one that's also not too bad as well now on their chemistry lines. That's more of an RBD, a similar RBD anti-spike one. Those would be the ones I would go after.
That's great. Fantastic. Let's go to the next question. And for those in the audience, please continue to submit your questions in the Q&A box. We have a live feed here. Next question, can we touch on the various subtypes of the coronaviruses being studied and or identified in transplant patients prior to COVID-19?
I mean, the 4 human coronaviruses, so, 229E, OC43, NL63, and HKU1, so 2 alphas, 2 betas, really have nothing to do with SARS-like coronaviruses and the sarbecoviruses. 2 of these actually weren't even found until after SARS sort of won in 2003. They really just weren't even looked at closely to figure them out. And actually, coronaviruses writ large are thought to have less chance of spreading to the lower respiratory tract. They're thought to be more benign. All told, they're probably like last on the list, maybe just above the rhinoviruses in terms of like respiratory viruses for which, medical types were looking for a vaccine. It basically goes flu, RSV, parainfluenza, metapneumovirus, and then you'd go down to the coronaviruses. So those are the ones that we were looking at before. As for, I don't know if Jason wants to talk about, or Emily want to talk about quote unquote subtypes of SARS coronavirus too, or if you even think there are subtypes of this thing at all. It depends if you're a lumper or a splitter.
I'll briefly talk about that, but as you can tell from Alex's, that, yeah, I think, we would, as the sort of mutational rate of coronavirus is actually very low compared to other RNA viruses. And although there's a lot of popular press about people worrying about coronavirus mutating into a new strain, I think, at UCSF, we know we have to be very careful about our terminology. And typically when we use the word strain with respect to these viruses, we're talking about a mutation that causes a phenotypic consequence, as opposed to, these things are going to accumulate mutations just by spreading by neutrality. And so there's so far I wouldn't say we would define anything as a different strain. We do definitely define different subtypes when we're, phylogenetically analyzing these sequences. But I think in the popular press, one particular mutation in the spike protein, this D614G that you may have read about, or DUG as it's sometimes referred to, has some minor evidence that it increases transmissibility, but I don't think you can fully reject neutrality yet. There's basically at this point we don't really have any evidence that that the coronavirus is mutating. We have not really even put any selective pressure on it for it to mutate to do anything, right? It sort of made the mutations it needed to do to jump to human, and it seems to be doing quite well.
That's great. I actually have a follow-up question going off of the strains and subtypes. Does that mean that there are multiple types of vaccines in development?
So I'll speak at the very high, the sort of, I'm sure Emily and Alex can answer at a deeper level, but, I am most of so there are definitely different vaccine approaches. You probably have heard of Moderna's mRNA vaccine, which is just like the mRNA of the spike protein. And then certainly there are sort of more traditional sort of inactivated virus vaccines being pioneered in other countries. So there are definitely different approaches. So far we haven't, in terms of sequence, had any sort of evidence that there's a particular sequence that's bad, that, that any of the sequences that are being used for vaccine candidates, there's nothing necessarily poor about them. Even that D614G mutation I mentioned that has sort of swept in its frequency, it has not shown different sort of reactivity to neutralizing antibodies and such.
Great.
Just add something to that, or actually really ask Jason's thoughts. It seems to me, from having kind of been a little bit on the outside of the genomic epidemiology that the Biohub is doing, when we have positive samples at the CLIA lab, if there's an agreement in place with the public health department that the sample came from, we send it to a group at the Biohub that just does whole genome sequencing. And so we've generated hundreds of genomes that way. And that's been used to kind of follow, follow spread from a public health standpoint and things like that. It seems to me that we're kind of in a sweet spot with this virus in terms of the mutation rate. I think if it was much faster mutating, we wouldn't be able to sort of pick up on these kind of trends with so much granularity. And if it was any slower, I think we would lose any relevant information about who's giving it to who. So I'm just curious, Jason, from your experience with viral evolution, do you think there's any reason why we're in that point? And is it possible we're going to get out of it? We're going to get a more higher mutator phenotype coming along by some polymerase mutation or something?
Yeah, I mean, I don't know. I mean, the virus, as Emily's kind of alluding to, is rare in that it has a proofreading polymerase. So it has a very low mutation rate. Has a very high recombination rate, but I don't think we're quite yet worried about recombining with something else. I think, yeah, in the sense that we have, in the sense that its mutation rate has been sort of this low enough that when we see real mutations, we can sort of do the sort of tracing that the sort of genetic sort of detective work to figure out what the transmission cluster was. I think it's been great in that way, but I don't know. I mean, we haven't added selective pressure to it, and as far as I understand, what's going to determine the efficacy of the vaccines are things that I'm certainly not an expert in terms of how the immune system is going to respond in terms of T-cell responses, and that I certainly don't know. And the experts that I do know are careful in their responses in terms of how they think protection is going to be conferred and how the virus might respond.
Great, thank you. Let's see here. Next question, and again, for those in the audience, continue to share your questions in that Q&A box. These all look fantastic. Could you speak to whether there is sustained immunity for those who have had COVID-19, and maybe a bit on vaccine efficacy and how that compares?
So I'm happy to take this one, but so I mean, we just recently put out a paper on neutralizing antibodies on a fishery boat showing protection in humans. That really is the first sort of data on that in humans. It's very limited. It was a boat that had an 85% attack rate. So if you showed 3 people who had neutralizing antibodies and they didn't get infected, that's statistical significance. So we have 3 people who have been infected. I also say we have most of the cases where you get called about reinfection are not really sort of good bona fide don't bear out reinfection. There's obviously these cases in Hong Kong and Europe. We've had a couple here too, where I'd say, okay, that was, I'll give it as like, okay, virus, you were there. But actually didn't really cause symptoms the second time around. And, it does look like we're seeing pretty good protections. I mean, you can't answer longitudinal questions without time, unfortunately. So it is maybe a little concerning that right now we're starting to see some of those cases where those stories pop up in sort of the end of August going into September, but we're really looking at people who are infected towards the end of February and early March. So, I mean, that gives us some idea of sort of an average bear sort of, time frame. Obviously, the titers of neutralizing antibodies that people get from different infections can be wildly different, can range really over almost 100-fold. And so that's a major issue. If you look at the titers, though, that we had, for instance, that boat study, and the titers that are being achieved by the vaccines in their sort of day 28, day 56 follow-up, right, where you're kind of at your tippy top titers. The vaccines look great. I sort of even go farther than what Jason's saying in terms of I don't really think that the diversity of the virus has really anything to offer as a problem for a vaccine or any therapy at this point. I mean, that's actually our friend right now. It's only had 10 months or so to evolve in the human population. So it's really quite sort of threadbare when it comes to mutations, and compared to when you compare to the other coronaviruses or other respiratory viruses writ large. So I don't think it's gonna be a major complicator. It's really just gonna be how long do those antibodies want to hang around, and which of these 7 shots or 6 shots on goal is going to be able to drive the longest, the highest titers for the longest amount of time from an IgG response, and we're just not going to know the answer to that, right? I mean, we only have data. I mean, people who got the Moderna vaccine from the beginning only got stuck basically in like March, and the first follow-up paper was like in May or June. So I mean, you're really only now getting sort of like 4 to 5 month levels on those individuals, and that's only like 15 people. So it's just going to take some time. I think the vaccines are going to work well out of the gates, and then the question is really going to be about longitudinal levels and how well we're able to actually get it together as a world to sort of roll out these vaccines, get the testing, and drive this thing as far down as we can to get it back to like SARS-1.
Great. We have a question here. Let's see. Maybe this can go to Emily. Would love to hear more about rapid tests, in-home tests, and the dilemma of FDA approval, perhaps the contention of limited sensitivity for diagnostic testing, but also good from a public health standpoint. Any thoughts on that, Emily?
Yeah, I think that's a great question, and something really important to consider. I think that there's a place for a variety of different types of tests with different levels of sensitivity. We just don't have enough data yet to know, because it hasn't been around long enough, truly what the sort of sensitivity cutoff would need to be to detect people who are going to be able to spread the virus. That's really I think the most important question from a public health standpoint, and we just don't quite know the answer. There's been some interesting modeling suggesting that, if you were to use a quite low sensitivity test, but test everybody every 2 days, for example, that you could have a similar effect to testing everyone less frequently with a higher sensitivity PCR test. I think that if that's true, that's great, and we need to come up with that low cost, low sensitivity test, and most importantly, we need to manufacture it in incredibly large quantities, and then of course solve the regulatory aspects around it as well. I think something that I've learned from this experience that I never would have really thought about before, or even questioned before, is how little, what a relatively small fraction of the kind of the marginal costs in terms of labor and resources of doing a test go into the sort of high-tech aspect of it, and how much of it goes into the pre-analytical, low-tech, receiving the, thanks Alex, receiving the tubes, unpacking them, entering the data. Serena totally knows what I'm talking about. Entering the data into the LIMS system, putting the sticker on the tube, transferring the liquid in the tube safely from a tube into a plate or into whatever system it needs to go into. And so I think it's once you've done all that, the RNA extraction and the PCR is kind of the easy part. So unfortunately, I don't feel quite as excited about these kind of new technologies that are going to solve all of our problems, without also the backing of a really strong investment in people and training, and bringing in the workforce that's needed to do these things and expanding lab capacity. I think that there is a possibility for a silver bullet somewhere out there if someone can really come up with highly sensitive true point of care test where either you literally just get the answer in front of you at home, or there's some process by which you yourself do the accessioning and send it into a lab already labeled and all that sort of stuff. Unfortunately, I haven't seen that appear yet, and I suspect that if and when it does appear, there are going to be manufacturing challenges and supply chain challenges as well, because just thinking of the scale at which you would have to deploy that for doing things like school reopenings. So those are my thoughts. I'm curious if Alex has any other thoughts there.
I mean, I think the only thing that people often, yeah, I completely agree. That was fantastic. And, you know, unfortunately, it's a horrid fundraising pitch, right? We need to train individuals because this is really hard and we don't have a magic bullet and talk to me in a few months, like, right? But that's the honest truth, right? That's why you go through all this health sciences education at UCSF, right? There's not a magic bullet. We can't do this with, you know, just take 18-year-olds and everything will be copacetic. I think the one thing that really comes through with these high frequency test approaches, or like damn the sensitivity, is that when you start to test at that level, you start to really, your specificity, you start to abuse your specificity, and then you start getting tons of false positives that people often just, you know, that need to be adjudicated. I mean, that actually even happens with the very good test, because like you said, the operations are challenging. I mean, it's really hard to run that many PCRs without having some false positives or having some, you know, queries on amplicon contamination and, you know, all the things that go into that. And so I think that one of the problems with the sort of like, you know, just the flood the zone approach, you know, is just kind of what it actually does to human psychology when people start just turning off and just not listening to the test, right? We've had a great intervention here with masks, right? That we've had right at the tip of our we've right here, you know, it's been available. And it really, look at how much effort it is just to like invoke that. And then, like you said, from manufacturing angle, I mean, a lot of the things, the analogy that we reach for probably the most when talking about these like high frequency $1 at home, it's going to be amazing. It's like, that's like floss, like, right? Like you need, like, that's like something that you ask people to do almost every day that has, you know, maybe a slightly better cost point, but has a manufacturing base. And that's what you're trying to make, a laboratory test, like basically like floss or like a toothbrush. I mean, something that you can basically, you know, it's just, and so those, we don't have the history, don't have the manufacturing chains. Those things might come in over the next like year. But right now, it's going to be really hard. And even if it does come, like even literally in the last few hours, there's this EUA for Abbott antigen test. Looks pretty good. I'm actually getting texted about it right now. I want to go read about it. But do I think that they can manufacture that in a period of 1 to 2 months? It's going to make, you know, much difference? I don't know. Like I'm sort of bearish on those sort of things. So, and even then, we're going to have a lot of PCR testing to adjudicate those false positives and false negatives, right? They really are screening tests for the most part. So, yeah.
Yeah, those are really good points. You could also think of, you know, what if we were, if everybody in the world was trying to take a pregnancy test every single day, you know, how would the supply chain of that go? Because people talk about that all the time. Oh, we have a pregnancy test, you know, why can't you just put it on a strip like that?
Right.
And again, that would be great.
And the rub with the pregnancy test analogy is the thing that makes a pregnancy test really helpful. One, pregnancy tests aren't great, right? It detects about a billion proteins is their limit of detection. We do an at-home pregnancy test, and they still cost $8. And the nice thing about pregnancy tests is they double every 48 hours, right? So like, that's a fantastic analyte, you know, right? You can come back and retest, and you'll have even more there, right? That's how I adjudicate. That's how I want to adjudicate my false positives or false negatives, to have that. And unfortunately, this virus is doing the exact opposite. You never see, you know, that's the big difference between SARS-1 and SARS-2 is SARS-2, you can't ever find the viral load go up. You only find it go down because it basically peaks in that pre-symptomatic time. And so you just, the next sample, it's always lower, lower, lower. So it's really hard to adjudicate those things. I mean, really, at some point, you're just like, screw it, let's do some serology.
Such interesting points. Thank you for that discussion. We have one question here. Let's see. Please let us know your experiences or thoughts about convalescent plasma. Anyone want to take that?
Yeah, so convalescent plasma has gotten a lot of heat recently, mostly because the FDA botched its messaging and because, you know, what is it, 70,000, more than 100,000 patients in the US have received it without any randomized controlled trials. I mean, let's take what we do know, at least from the very least. I mean, not about SARS-CoV-2. I mean, convalescent plasma is the original Nobel Prize in medicine, right? So like, you know, this is like 1900 stuff with the diphtheria toxin for the most part. So it can work. It really, the problem becomes defining the titers and the time point at which you need to give it. And I think that's where, you know, when you look at the data that Houston Methodist submitted and a few others, you know, really just finding that cutoff of the titers that you want to be able to get it, and then also like the timing. I mean, that's really, so I think most of the debates in convalescent plasma come down to those questions, and especially as we're getting the assays up online, it turned out that with this virus, because of that, you know, really 100-fold difference in in titers between different individuals, that really does, you have to be able to measure something accurately that's gonna tell you the efficacy from that angle. So I think that there is going to be, I actually don't think the FDA was wrong at this point to offer an EUA to convalescent plasma, but I would like to see, a little bit. I mean, I think you really want to push and try to just go to the tip top of those titers, and then try to consider doing it a little earlier. Like, those are the major considerations of why one trial looks good and another trial doesn't look good. And so I think that given our 120-year experience with convalescent plasma, it's not unreasonable to look at some of those subgroups and sort of use that data appropriately, rather than requiring another, the next, or like a 4 or 5 month RCT at this point, given the burden of the virus. But that's just a personal opinion. I would hate to have to make policy based on that.
If I can steal a question in here for you, Alex, what are your thoughts on monoclonal antibodies as being a stopgap before a vaccine, or because presumably the active ingredient in the plasma, right, is some sort of antibody. Right.
So it's gonna be fantastic. I think you see the mutations you see in the spike protein are very limiting, so you don't have to worry about what you usually have to worry about with monoclonal antibodies. You got to show it. I mean, obviously they sort of wrecked Ebola. They did a great job. It's very bullish for them. You've got Regeneron, you've got Lilly, you've got Vir, you've got a lot of different players sort of in this market. I'm optimistic. But they still got to do the trials, and I think that the hardest part is just finding the right population to do the study in, because you often don't get I mean, the thing that sucks about SARS coronaviruses is that they give you that 6 to 7 day incubation period, followed by 7 to 8 days when you're symptomatic before you actually present to the hospital. So you can actually be pretty late. I mean, by the time you look at someone who might need therapy, you're actually pretty late in your course. And this is what always determines the efficacy of respiratory virus therapy when you're dealing with lung tissue damage. And so identifying those individuals that you need to give a therapy where you're actually going to show an effect size, as opposed to someone who could have just done well at home anyway, is a difficult one, especially as you're seeing more and more cases in 20-year-olds and 30-year-olds. That sort of complicates some of the trials. That said, compared to the other viruses where we've recently gotten a lot of activity in for Zika and Ebola, this virus is not running away from us, right? We're still having 30, 40,000 cases a day here, there, and everywhere. So we can enroll, and it is possible to actually do good trials quickly with this virus. But certainly the changing demographics make it a little bit harder. But yeah, I'm pretty bullish. I think they'll do great. You just got to show it.
Yeah.
Thank you. We have another question here. What about this virus allows for so many asymptomatic cases?
It's $1 million question. I don't know the answer. I don't know if either of you guys have any insight.
That's the grant that I'm working on. No, it absolutely is the question. I think if you were to nobody has an answer for it, right? It's a great question. SARS-1, SARS-2, 80% by nucleotide. One made it to 8,000 cases. One is going to make 80 million, 800 million, right? So it's going to be it's definitely the key question. It's the feature that separates sort of the adults from the children in virology. The herpes viruses do quite well because they have asymptomatic transmission through and through. HIV did quite spread well in the human population because of not only pre-symptomatic but still a long period of asymptomatic transmission, and that's really what allows viruses to get. It's just such a key question. You look at the coronaviruses for this one between SARS-1 and SARS-2, where we actually have a great anchor. You have a 10 to 15-fold stronger binding to the human ACE2 protein in SARS-2. You have higher viral loads in the upper respiratory tract, which SARS-1 did not do very well. It's mostly lower respiratory tract. And it peaked at 6 to 11 days. So I don't know that's sort of I don't know if that's an explanation, it's sort of just a correlative, but you could be receptor driven. And of course, the coronaviruses devote 1/5 of their genome to screwing with interferon. They devote a lot of real estate to innate immunity antagonization. We just put out a paper on ORF6, which seems to shut down nuclear cytoplasmic transport because RNA viruses don't have to care about the nucleus at all. So they can just cut off traffic between the nucleus and the cytoplasm, and they get more ribosome for them. They shut down the interferon response. Everything's hunky-dory for the virus. And if you look at the SARS-1 and SARS-2 proteins for there, actually we found a greater effect of the SARS-2 protein. But the problem is there's like 10 different proteins that do that in these viruses. So you kind of have to like do all the additive effects of like, how does it all work together? And presumably that could be a potential effect too, if you can shut down the interferon response and really never allow the innate immunity to get going while you're trying to get up to 10 billion copies per mil in the respiratory tract. But I'm curious what other people think. Those are the two best explanations that I've been able to come up with.
I mean, yeah, I agree. It's the $1 million question. I think there are, I think there are interesting leads in there. I think there's, questions about, when we start going down to the basic biology, why are young people not affected, right? There's got to be some mechanistic reason, and it also in the sort of sex differences between males and females. And so I think, that's going to be not just Alex, there's going to be a lot of grants trying to figure this out. And just to emphasize to the audience, I think even, coming from a genomics perspective, even when we have the genome, there's been a lot of time spent on finding what are all the protein products, right? It's not straightforward like the human genome. Well, I wouldn't say the human genome is straightforward, but it's not as straightforward as one might hope. And there's a lot of basic biology to be done here. And I think, people at UCSF, a lot of labs have been working on that. And yeah, eventually the immune system is going to be involved somehow. I think the, circulatory system also, but I don't know.
That's great. I think we have time for just one final question. Let's take it back to the testing. Emily, do you think you can just go over just the different tests that are available, which are the most accurate, and perhaps which are the most reliable that a clinician might recommend?
I don't know if I can speak to that from what's reliable from a clinician standpoint, but I can just describe that the test we run at the CLIA Hub is LDT, a lab developed test. It's essentially just extracting RNA the same way that you would do in a research lab, and then using like an NEB RT-PCR kit to detect both the E gene and the N gene of the virus. We actually have a paper just out on bioRxiv showing that we came across a mutation in our N gene forward primer site that decreases the sensitivity of that assay by about 6 to 8 CT units, and that can cause false negatives if you're looking only at that one gene. So I can definitely say that assays that rely on only a single gene product are not good. We shouldn't be focusing on those. There are a lot of sort of sample to answer tests, like the Hologic Panther is everyone's favorite one right now, it seems like, and that's a little bit of a it's a little bit of a black box from an assay development standpoint. But it's RT-PCR. There are also isothermal amplification tests with LAMP assay. There's a few EUAs for CRISPR-based detection after isothermal amplification. From a research perspective, having done a little bit with isothermal amplification in the lab, it makes me nervous that those reactions can run away, and you can end up like taking your sample out of the freezer 3 weeks later, and it's gone from barely positive or not positive to roaring positive, and you're like, how did this happen in the freezer? I don't know. That gets you to start really worrying about cross-contamination and false positives. In addition, I think my research was on CRISPR-based diagnostics before this, and it's a little bit sad to me that I'm not able to use that in the work I'm doing now. But there's kind of like PCR is kind of the classic, and there's kind of nothing better than it right now. It's a little bit sad to say that, but I hope there will be in the future. But the tried and true is really what we're relying on right now. And then Alex mentioned antigen-based tests. I think those are really important to start looking at as they start getting better and better. And And that's what I've got to say on that.
That's great. Thank you. PCR, our bread and butter.
Yeah.
Well, I think that we are out of time here. Thank you to you all, Jason, Emily, and Alex, for sharing your stories and your experience with us. And thank you to our alums in the audience for submitting these questions and for tuning into this such a special virtual experience, highlighting the strength of the UCSF community. We'll end the evening here. Thank you so much and have a great night.
Thanks, Serena. Bye, everyone.